So today's topic will be glycolysis. And the word might seem scary at first, but you have all the skills you need at this point to understand what it means. Literally, glyco means sugar. Lysis comes from the Greek of loosening, or holding less tightly. We think of it as breaking today. So this is the breaking apart of sugars, right? In particular, glucose. So on the first slide here, I have 75% of what you need to know is in that figure on the right. Now, this semester is a little different in that we're doing it remotely and you don't have to come to class for the test. We're going to take it online. So obviously you're going to have access to these slides, this video, and everything during the test. However, as you learned from the previous test, you can't spend your whole time rewatching things or looking up something during the test. So it might be useful to know it ahead of time. So on the right here shows glycolysis in all 10 steps. The molecules on the far right are the structures you're going to learn. The things in black bold font are the names of those molecules. The things in blue are the names of the enzymes that can do those steps from one to the next. And there's several cofactors involved in there as well. You see it with the arrows. Now, I don't care if you remember if it's stage 1, stage 2, stage 3. That's kind of irrelevant. But I do need to know the steps and in order. And I'll tell you a brief story to help you remember it. Right? You can put it in your own words later so this makes sense. So we're going to start at the top with glucose. Right? So this whole story is going to start with just glucose. So if you don't remember how to draw glucose, that might be something you want to do before you move on with the rest of this lecture. So to remind you, we drew glucose before, as shown on the top left, in two forms, either in its ring form or in its straight chain aldehyde form. And you remember how to go back and forth. It's just forming a hemiacetal, or opening the ring. Right? So it still has one side of unsaturation either way, either the ring or the double bond. So we're going to start with glucose. And the first thing that happens is we're going to put a phosphate on it. Okay, the enzymes that put phosphates on things are called kinases. They transfer a phosphate on. This is one of your 6 categories from last time. We learned all our categories of enzymes. There's transferases, which take something away from one molecule and give it to another. That's exactly what we're doing here. We're taking a phosphate from ATP, which has 3 of them, leaving 2 behind. That molecule ADP can leave. And that one phosphate we took away, we're putting on carbon number 6 of glucose, which is at the very top of the ring structure, or the very last carbon on the straight chain structure. And if you look at the figure on the right, you see that the carbon number 6 at the top now has a phosphate on it, the PO3 with the 2 negative charges. Okay, so in the first step, simply, we're transferring a phosphate onto number 6. Okay. Then in the next step, we're going to convert glucose into its cousin fructose form. Right, so to do that, we have to open it back into its straight chain, so it looks like the molecule in the straight chain form again, except there's a phosphate on number 6. And we're going to turn the top of the molecule from an aldehyde into a ketone. In other words, we're going to move that double bond from the first carbon to oxygen down to the second carbon to oxygen. Okay, you've done this reaction before. We did it when we studied carbohydrates, so it's nothing new. So we're just going to turn glucose 6-phosphate into its cousin fructose 6-phosphate. And then right when we're done with that, that enzyme is going to wrap it back up in its ring form. Remember, fructose only having 5 members in the ring, not 6, because the carbonyl is now on number 2. So when it does the hemiacetal, or in this case hemiketal formation, it's attacking at carbon number 2, where the carbonyl is, not number 1. Okay, so we've done all this before. And you look on the right in the picture, you see fructose 6-phosphate. It's in its ring form. Carbons numbers 1 and 6 are sticking off the ring, just like they were before when we studied fructose. And number 6 still has its phosphate. In the third step, we're going to take that fructose 6-phosphate and add yet another phosphate. So this is another kinase, another transferase. An ATP is going to donate a phosphate again, leaving as ADP. Right? And that phosphate that was donated becomes the phosphate attached at number 1 now. So now I have a fructose with 2 phosphates on it, one on number 6 and the new one attached at number 1. Drawn in the figure, you see is a phosphate on numbers 1 and 6. Okay, so the first 3 steps are rather simple. You can tell yourself a story really quickly. We're going to start with glucose, phosphorylate it on number 6. Then we're going to turn it into fructose, still has the phosphate on 6. Then we're going to add yet another phosphate, this time on number 1. So now I've arrived at a fructose with 2 phosphates on it, on number 1 and 6. It's a really short explanation of the first 3 steps. Okay. The next step, the molecule opens back up into its straight chain form. So now we have a fructose. Looks a lot like glucose, except the carbonyls on number 2. And we have phosphates on both CH2OHs, number 1 and number 6. Okay. And this molecule is basically going to get a divorce from itself. It's going to be cut equally in half. Each side is going to get 3 carbons. Each side is going to get a phosphate. Each side is going to get the same number of O's, the same number of H's, and so forth. So it's going to make 2 molecules that are isomers of each other. So if you take a 6-carbon fructose with 2 phosphates and you cut it in half, the 2 pieces, since they're equal to each other, right, they're isomers of each other, are each going to have 3 carbons and each going to have 1 phosphate. It's pretty easy to remember that. Right, so shown in the figure on the right, you'll see the 2 pieces. We'll cover the details more as we get into the lecture. But you see we have 2 3-carbon molecules. The one on the left is a ketone, still has a phosphate. And the one on the right is an aldehyde, still has a phosphate. So they look almost the same as each other. In fact, they're isomers of each other. The only difference being where the carbonyl is. Okay? And the next step, the 5th step, we can simply convert one to the other. I can turn one aldehyde into the ketone or the ketone into the aldehyde. You can see this is analogous. It's almost identical to step 2 back there where we turned glucose into fructose. You're doing the exact same chemistry. It's just on a smaller substrate this time. Okay, so that's the first 5 steps. We're halfway done, right? We've covered half of glycolysis in under a few minutes. So let's continue our story. We took glucose, we phosphorylated it. On number 6, we took the glucose 6-phosphate and turned it into its cousin fructose 6-phosphate. That's an isomerase. Right? We didn't add or remove anything. We just made an isomer. We added a 2nd phosphate to make fructose doubly phosphorylated on 1 and 6. That molecule opens up into a straight chain and gets a divorce from itself, equal divorce. Right? So you get 2 3-carbon molecules, each with 1 phosphate. And those could be interconverted from one to the other. Right? And that's half a glycolysis right there. Right? So what have we invested so far? Well, so far we've added a glucose, and a couple ATPs have donated a phosphate. So we're in the hole for 2 high-energy phosphates at this point. So if you think of it in terms of how much energy or ATP am I going to get out of this thing, or what products am I going to get out of glycolysis, so far I'm in debt for 2. Right? We must recover that in a minute. Right? So steps 6 through 10 are only drawn once on the page, but you realize we have 2 copies of that molecule. We have the glyceraldehyde 3-phosphate on the right. That's the aldehyde piece with 3 carbons. And we have the dihydroxyacetone phosphate on the left. If you remember, those are the 2 3-carbon sugars minus their phosphates I had you remember for the carbohydrates lecture. One could be converted into the other. So when I continue with the next part, I essentially have 2 of those molecules, right? I have 2 glyceraldehyde 3-phosphates that continue with what's labeled as stage 3 here. That's why there's the big 2x in the bracket shown there. It doesn't mean one molecule is going to go through this twice. It means there are 2 molecules that enter this stage. So I'm going to enter with 2 glyceraldehyde 3-phosphates, and I'm going to finish it up with 2 pyruvates. Okay, that's all that means. It's just showing it once. If you want to draw another copy of those molecules right beside themselves, that would be more accurate. Just you want to see where the carbons end up. So starting with the next reaction, reaction number 6, we're going to take the glyceraldehyde 3-phosphate, and we're going to do 2 things at once to it. Okay? It is an aldehyde, so I can oxidize it. This is the only oxidation step in this entire pathway. Right? So it's the only redox reaction in all 10 steps. If you take an aldehyde and oxidize it, you're going to get a carboxylic acid or a carboxylate when it loses its proton. Right? At the very same time, I'm going to use a phosphate I pick up off the ground. Well, it's inside the cell, so I guess I pick it up out of the air, right? And attach it to that same carboxylic acid. So I'm turning the aldehyde into a carboxylic acid, that's oxidation, and I'm going to attach a phosphate. Right? So those 2 things are going to happen simultaneously in that step. Right? So that's step 6. It's glyceraldehyde 3-phosphate dehydrogenase, which gives away that it's oxidizing it, but it's also attaching a phosphate. From where did I get that phosphate? Not ATP. I just picked it up out of the air, right, or out of the cytoplasm. Right, so all this is taking place in the cytoplasm, and it just grabs a phosphate floating around. There's lots of them. Okay, so I'm in a similar situation that I was back with step 3. Right, I have a molecule with one phosphate on it. I added a second. I have two phosphates on it now. So much like the fructose 1,6-bisphosphate I had above, I have a 1,3-bisphosphoglycerate. So it's another 3-carbon molecule with two phosphates on it. Okay. At this point, I'm going to take one of those phosphates off. In fact, it's the very one I just put on. That bond that's holding it there is not very stable. It's got a lot of energy built up in it. Right? You can think of it that way. So it's readily remove the phosphate. So we don't just let the phosphate go. That would be pointless. We didn't get anything out of it. We're going to give that phosphate to ADP. Right? So an ADP molecule accepts the phosphate from 1,3-bisphosphoglycerate. Right? That leaves it with 1. It becomes 3-phosphoglycerate. So clearly it took the 1 off of carbon number 1. That's at the top of the figure. And it gave it to ADP to make an ATP. So at this point, how many ATP have I invested? Well, that would be 2 in the beginning, at the first and third steps. And how many have I recovered? Well, you might think 1. But keep in mind, we're doing that step 6 I just described and step 7 I just described for 2 molecules that both came from the glucose. There's 2 copies of this at this point. So actually I've recovered 2 ATP after step 7. So at this point it's kind of break even. I invested 2 and I've recovered 2 after step 7. I still have those 3 phosphoglycerates sitting there, and more importantly, in fact, I got 2 NAD pluses converted to NADHs, right, which is worth way more than 1 ATP. We'll see later. Right, so to do that redox reaction of the glyceraldehyde to the glycerate, I had to use an NAD plus, and it converted to NADH. That's an oxidation of the molecule and reduction of NAD. And now I've broke even. I got 2 ATP out of it. I'm not done though. I have the 3 phosphoglycerates sitting there. And that's a rather stable molecule. I want to make it unhappy with me, right? Do something to it to make it a little less stable. Can you see from the figure on the right that the carboxyl group at the top, it's drawn as a C with 2 oxygens and a negative charge. Either one could be the double bond and either one could be the O minus. That's all that means. So there's a negative charge at the top of the molecule. And there's also some negative charges at the bottom of the molecule where the phosphate is. And they're about as far from each other as they can get in that molecule. That's stable. Taking negative charges and putting them as far from each other as they can, that's a stable thing. So what I'm going to do in the next step is take the phosphate off of carbon number 3 and move it up to number 2. I'm just going to switch the H and the phosphate, and phosphate's on number 2 and the OH will be on number 3. So I haven't really done anything interesting. I just moved a phosphate from number 3 to number 2. But I did get those negative charges a little closer together, so that's less stable. Okay? And the last thing I'm going to do before I get some more energy out of this thing is I'm going to remove some water. You've done this reaction before, right? The dehydration stuff. If you look at that 2-phosphoglycerate on the right, you notice that carbon number 2 has a phosphate on it. It also has an H, because carbon has 4 bonds. The carbon number 3 obviously has 2 hydrogens and an OH. I'm going to remove the H from number 2 and the OH from number 3. That's going to leave as water. And you've done this type of reaction before. You're turning an alcohol into an alkene, right? Dehydration. This is a lyase. So enolase is the name of the enzyme. And now we have a double bond between carbons 2 and 3. Well, how's that any different? Is it less stable? Is it more stable? It's quite a bit less stable, because now that double bond prevents the rotation of those 2 negative charges away from each other. So this is a very unhappy molecule at this point. It would love to resolve the situation, and we let it. So we let the phosphate go. We don't just let it fly off into space. We do what we did back up at step 7. We take the phosphate off and hand it to an ADP, making an ATP. And then the double bond can rearrange, but from going from between the 2 carbons to the carbon and oxygen, making a carbonyl, and you end up with pyruvate at the end, which is very stable. So in the end, what did I get out of this? Well, after step 7, I got back 2 ATP, which replaced the 2 I invested. And after step 10, I just got 2 more, because remember, there's 2 copies over there. So overall, I invested 2. My output, my gross output of ATP was 4, which is a net gain of 2. Okay, and again, I got 2 NADHs back in step 6. And I have 2 pyruvate molecules, which we'll see what we do with those on our next lecture. Okay, so that's a brief rundown of the story. Let's tell the story again in the shorter words and see if you can get the framework in mind, and then all you can do is fill in pieces later. We start with glucose. We're going to phosphorylate it on number 6. Turn that into, start with glucose. I don't know if I said fructose. Glucose, phosphorylated on number 6. We're going to isomerize it to fructose 6-phosphate, add a second phosphate, so now they're on 1 and 6. The molecule is going to get a divorce from itself into 2 3-carbon molecules, each with a phosphate. One is going to get converted to the other, so I have 2 copies of the aldehyde now, and I'll talk about what happens to one of them. That aldehyde gets oxidized to a carboxylic acid, and a phosphate gets put on number 1 at the same time. In the next step, we take that phosphate right back off and give it to ADP. Then I have 3-phosphoglycerate. I move the remaining phosphate from number 3 to number 2. I remove a water molecule to introduce that double bond. It's really unstable now. And I take the last phosphate off, give it to ADP, making the last ATP, and I have pyruvate. That's the really short version of the story. If you can get that in your head and then fill in the details on each step you want, it'll help you learn this. Okay, so let's go through these steps one at a time and talk about the details I want you to know. Okay. So step 1, hexokinase, it puts that first phosphate on glucose. So looking at the left, at the glucose molecule in its ring form, there's quite a few OHs on the thing, right? There's an OH, there's an oxygen virtually on every carbon, and there's 5 OHs available to do something. The 6th OH is now in the ring, right? So there's 5 OHs available, number 1, 2, 3, 4, and 6. Which one do you think of those 5 is the most accessible, right? One of the 4, numbers 1, 2, 3, 4 that are actually on the ring, or number 6 that's sticking off the ring? Which one do you think the enzyme would have an easier access to?
Number 6.
Exactly, and the reason is it's farther away from the other oxygens. Right? So exactly number 6 is where we're going to put it. If you don't just remember number 6, just look at the molecule and ask which one of these can I throw a phosphate on the easiest without having to interact with the other oxygens very much, and that's number 6. Right? Keep that in mind for my question coming up in a couple slides. So this enzyme, hexokinase, uses ATP as the phosphate donor and leaves ADP as the product. So don't think of ATP as a ball of energy. I know they like to do that in like freshman biology classes. It's not some energetic ball of energy, right? It's a molecule, right? And it has 3 phosphates on it. We're going to remove the last phosphate, the gamma phosphate, alpha and beta and gamma phosphates. We'll take the last one off. We'll put it on glucose's number 6 OH, and we have ADP left over. So why would a cell do this right away? As soon as the glucose enters the cell, this happens. Well, let's compare the 2 molecules. Glucose, can it pass through a membrane? Well, not really. It's got a lot of OH groups, it's rather large, and it's quite polar. So it's not going to easily pass through a membrane. However, glucose 6-phosphate is almost never has a chance to get through the membrane. It's got that negatively charged, doubly negatively charged phosphate on it now. So if glucose even had a prayer to get through the membrane, glucose 6-phosphate doesn't. There's no chance of it leaving the cell. So this traps glucose in the cell, which is a good thing because we're going to get something out of it. So do I want to regulate this step? Sort of. Right? I don't want it to go crazy and phosphorylate every glucose I could possibly throw at it, but any that get in the cell, throw a phosphate on it. I don't mind spending that ATP because it's my glucose now. I'm never going to lose it. And I'll get more than one out of it later. Okay, so in the next step, at the top of the screen, we take glucose 6-phosphate in its ring form. We open it up into the straight chain, and you guys know how to draw glucose at this point. Remember the OHs for numbers 2 onward go right, left, right, right. And you see the picture in the middle there is glucose in its open chain form. We simply move the carbonyl from number 1 down to number 2, and now we have fructose. The phosphate's still on number 6, it wasn't involved. And then we wrap it back up in its ring form, and we do that, we get the attack of the last chiral OH on number 5 to the carbonyl carbon again, but it's on number 2 this time, so we get a 5-membered ring. Right? Moving on to step 3, answer the same question I asked earlier. I'm going to put a second phosphate on. Well, I certainly can't put it on number 6, right? It's got a phosphate already there, and it's a -2 charge. the new phosphate doesn't want to get anywhere near that other negative charge. So my options are the OH number is 1, 2, 3, or 4. Number 5 is still in the ring, so it's inaccessible. Which one, 1, 2, 3, or 4, looks the easiest to get to?
1.
1, for the exact same reason you told me earlier. It's the one off the ring. Well, number 6 is off the ring, but it's got a phosphate on it already, so let's stay away from that other charge. So this enzyme, phosphofructokinase, what that means is it's a kinase, which transfers phosphates, and the phosphofructo part tells me the substrate. It's an already phosphorylated fructose, so a phosphofructose. So we add another phosphate to it. ATP is again acting as a donor. This is a different ATP, right? And now I have a fructose with phosphates on numbers 1 and 6. So in the terminology, you'd think it'd be called diphosphate, and that's perfectly fine. But we tend to use bis when we're talking about biomolecules in place of di. So bis means exactly the same thing as di. So fructose 1,6 bisphosphate. This reaction is kind of not the weakest link in the pathway, but the most important and the one that controls the whole rate of the pathway. If this one's not working, the whole thing doesn't work. And it's also our committed step. At this point, I can't do anything else with this molecule. Once I turn it into fructose 1,6 bisphosphate, I need to finish this pathway, right? I let the first step 1 go. I know it cost me an ATP, but I let it go, thinking from the cell's point of view. I let it go because it trapped the glucose in the cell. I can use it for later. Now I'm investing a second ATP. This is getting expensive, and I'm getting nothing out of it so far. So if I do this step, I want to finish the pathway. So this is the committed step for glycolysis. And it's also the one that sets the pace, and it's the most important regulatory step. We'll talk about that at the end. Okay. So I have a fructose in its ring form with 2 phosphates on it now, and it's going to open back up into the straight chain one more time. This is the last time we have a ring. When it opens to the straight chain, it's going to look just like the one above, except there's going to be a phosphate on number 1 as well. Okay, so that looks like this on the far left. There's your 6 carbons of the fructose, and phosphates on numbers 1 at the top and 6 at the bottom. Sorry they switched colors on you, but 1's blue at the top and 6 is now pink at the bottom. All right. The next step, we're going to take that molecule and it's going to get a split from itself, an equal proportion of atoms. So each side's going to get 3 carbons, each side's going to get 1 phosphate, each side gets the same number of oxygens and so forth. So color-coded there, you have to split it up a little differently because the top 3 carbons have a double bond in them in that carbonyl. The bottom 3 carbons don't, which means one has 2 more hydrogens than the other, right? The bottom one doesn't have that site of unsaturation, so there are 2 extra hydrogens on the bottom. So if the bottom were to give one of those hydrogens at the top, it's labeled in blue, then we have an equal split. All right, so that's what happens. The bond in black is the one that breaks. The hydrogen goes with that carbon, and we end up making an aldehyde on the bottom. So we have dihydroxyacetone phosphate on the top and glyceraldehyde 3-phosphate on the bottom. Even split. All right, this enzyme is totally reversible. We could go backwards. But if we split the 2 apart, I can convert one to the other. Right, and that's shown at the bottom. The dihydroxyacetone phosphate can easily be converted to the glyceraldehyde form. This is identical to back at step 2. Let me go back and show you real quick. Step 2, if you look at the top, we turned an aldehyde into a ketone, or we could go in reverse and turn ketone to aldehyde. All we're doing is moving that double bond from carbon 1 to carbon 2. Here we're doing the exact same thing. We're moving the double bond from number 2 to number 1. The only difference here is this molecule is smaller. It doesn't have 6 carbons anymore, it only has 3. Doesn't really make a difference for the enzyme. It's doing the same chemistry, it just needs to recognize its substrate properly. Okay, so now I've turned the dihydroxyacetone into a second glyceraldehyde 3-phosphate. Right, so when I did aldolase in step 4, I got one of each, and I can convert the top one to a second copy of the bottom. So I have 2 of those glyceraldehyde 3-phosphates at this point. I haven't recovered anything from this reaction so far. I've done 5 steps, and I've invested a couple ATPs. That's all I've done. I need to get something out of this now. So the next step, this is the only redox reaction in the whole pathway. We're going to take the glyceraldehyde 3-phosphate. Keep in mind we have 2 of these. I'm only going to show it for one of them. And we're going to oxidize it. And if you remember, aldehydes oxidize to carboxylic acids. So we're going to oxidize it to a carboxylic acid and at the same time add a phosphate. Well, those two reactions, one is favorable. Oxidizing it to a carboxylic acid is a favorable reaction. Adding a phosphate is not favorable. Can you imagine why? The glyceraldehyde 3-phosphate already has a phosphate. It's got negative charges. Adding some more negative charges to this molecule doesn't seem favorable, right? You don't want them that close. So I'm going to use one reaction, which is favorable, the oxidation, to drive the other one forward. That's why we coupled them together in step 6. So that's shown here in this diagram. If you look at the first line, it shows the oxidation step. I turn the aldehyde into a carboxylic acid. And if you remember from when we did the oxidation and reduction of aldehydes and ketones and alcohols, that you get the other oxygen, the other OH from a water molecule. Right? So there it's shown on the screen. If I just did that step, that would be the end of the reaction. It's a very stable product. And in putting the next one on is not favorable at all. You can tell by the little arrows there. It prefers not to do that. So I couple them together so they happen at the same time. So the little diagrams at the bottom, if you remember from our enzymes lecture, the y-axis says free energy. The x-axis said reaction progress. Not necessarily time, just as the reaction proceeds. So as the reaction proceeds, we have an activation energy. You see the little gray hump we have to get over? Then it falls down in this deep pit. To continue, it would have to get out of that pit, which is not favorable. That's not energetically possible. So what this enzyme actually does is raise the depth of that pit. It doesn't make it as deep, right? And it's shown on the right. The middle pit isn't as deep because the enzyme's holding on to this intermediate. Right? You don't have to know all the chemistry behind it. I just want you to know that the enzyme is taking a favorable reaction, the first line up there, and coupling it to an unfavorable reaction, the second line. Overall, it forces it to do both or none. That's what this enzyme is doing. Okay, so we get the oxidation step and the phosphate put on, or neither happens at all. Right, so this is a very common thing to couple two reactions, use one to drive the other. So ultimately, we've taken our 3-phosphoglycerate and turned it into 1,3-bisphosphoglycerate. Okay? What do we get out of it? Well, we didn't use an ATP, so where'd the phosphate come from? We picked it up out of the air, out of the solution, out of the cytoplasm. Right, so the phosphate's just floating around, and we pick it up and put it on carbon number 1. Right? The other thing we get out of this is an NADH, which is very important for later. Right? It's actually way more important than the ATPs we get out of this. Right? So we turn glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. We consumed a phosphate from solution, not an ATP, and we got an NADH out of it. All right, so we put a second phosphate on. We're going to let it come right back off. Remember, this wasn't favorable to throw it on here. But we're not just going to let it go. We're going to capture it. Right? So the next step, we take that 1,3-bisphosphoglycerate off of the molecule and give it to ADP. That's what this enzyme does. This one's totally reversible. Right? So this is not much difference in energy here. We take the phosphate off, give it to ADP, which makes an ATP, and leaves us with our very stable 3-phosphoglycerate. That's the very thing we were trying to avoid back here in that first line. We didn't want to stop there. There was some energy left in this, so we captured it as an ATP. So to answer my question here, which I asked earlier, how many molecules of ATP have been formed at this point?
2.
2, right? Because it looks like 1 up there, but remember we did this twice for 2 of these 3 carbon molecules. And how many have been used or invested at this point?
2.
2. Which steps were those? So think back to the beginning. Where did we use or invest ATP? Which steps?
1 and 3.
1 and 3, exactly. The kinases, right? Where we put the phosphates on glucose or on an already phosphorylated fructose. Okay, so at this point we've broken even, right? I have 3 phosphoglycerates sitting there. I got my invested ATPs back. I did get that NADH, 2 of them in fact, but I can continue. There's still more energy here. Okay, so the last 3 steps I'll cluster together here. We take the 3-phosphoglycerate on the left, right? It's a fairly stable molecule. The charges are as far apart as they can get. And I move the phosphate from number 3 up to number 2. Pretty simple. That's an isomerase, right? It's classified specifically as a mutase because a mutase is an isomerase that moves something bigger than a proton or electrons. So it's moving a whole phosphate. So I put it up to number 2. The negative charges are a little closer together, so this is not as favorable. And then I remove a water molecule, right? The water comes from the OH down on number 3 and the H on number 2. And that puts a double bond between carbons 2 and 3. This is dehydration. You did this before, right? And now that puts the carbon at number 2 into a different geometry, right? Its geometry, instead of being tetrahedral like it has been for a long time, now it's what? In the phosphoenolpyruvate, what's the geometry of carbon 2 there? This carbon. So it was tetrahedral before. What is it now?
Trigonal pyramidal?
Trigonal pyramidal would imply it has a lone pair, right? Does carbon have a lone pair here?
No, it's plain.
Trigonal planar, exactly. So, I mean, it's not a bad guess. You didn't say linear, which is wonderful. But remember, carbon here doesn't have any lone pairs, so it's trigonal planar. So the carbon number 2, carbon number 3, carbon number 1, and the O next to the phosphate over there are all in the same plane, right? As well as the 2 hydrogens at the bottom. But all 6 of those atoms are in the same plane. So whereas before we moved the negative charges closer to each other, they were still in a tetrahedral arrangement. They can get away from each other. Now they're restricted to being in the same plane. So these charges really don't want to be this close together. And we also have another problem. It's called a beta-keto arrangement. And the easiest way to resolve this is for the phosphate to just leave. And that was our whole goal, is to get this phosphate to want to leave. Right? So it's favorable for this phosphate to come off, but we don't just let it go. The next enzyme, pyruvate kinase, takes the phosphate off, gives it to ADP to form ATP, and we're left with pyruvate, which is very stable. Okay, so how many ATP have I made just from step 10?
2.
2 because I did it twice, right? I got 1 from step 10, but I did step 10 for 2 different molecules. You're absolutely right. And I get 2 ATP from this step. So overall, I've gained a net of 2 ATP. I invested 2, I got a gross of 4, a net of 2. Okay? So looking back at these reactions, you can tell me which ones were reversible or which ones were irreversible. So if we really quickly go back to the beginning, just look at the list. All of these reactions are completely reversible except for 3, right? And they should be easy to pick out. Right? Which ones do you think are completely irreversible? There's only 3 of them. And I'll give you another hint. They all involve an ATP. So at the top, we have step 1, where we put the phosphate on glucose. And step 3, where we put the second phosphate on the already phosphorylated fructose. And then at the very end, at step 10, where we took the phosphate off of phosphoenolpyruvate to make an ATP. Those 3 are irreversible. You notice those are the single arrows in this diagram. All the rest are double arrows. Right? So every step here is completely reversible except for 1, 3, and 10. Okay, so looking at these overall, if I write an equation for all 10 reactions put together, we started with a glucose. We did invest ATP, but we got, we invested 2, we got 4 out of it, so we only draw the net in the equation here. So I'm getting 2 ATP made from 2 ADPs and phosphates. I also converted 2 NADs into NADHs. I got 2 water molecules out of it. And my glucose, all 6 carbons of glucose, are now found in the 2 3-carbon molecules of pyruvate. So this is the net reaction of the whole thing. I get 2 ATP, I get 2 NADH. We're not too concerned with the 2 water molecules. And all the carbons of glucose are now found in the 2 pyruvates. What I want you to get out of the table at the bottom is it helps you remember which ones are reversible or not. If you remember back to the enzyme kinetics discussion we had. Delta G tells me if a reaction is what? What can delta G tell me about a reaction? So, you guys remember, we had delta G, it could be positive or negative. What did that mean?
If it was spontaneous or not.
So it's spontaneous if which one is true?
Delta G is negative.
If it's negative, exactly. So looking at all these on the, let's just pick one. Let's do the delta G on the far right. So it says the first step is negative 33.5 kilojoules per mole. That's the delta G. I'm not so interested in the units, but looking at it, it's a fairly large negative number, right? That's number one. We know that's irreversible. We can say the same for step 3, and we can say the same for step 10. It's a rather large negative number. Look at all the other steps. Not 1, 3, and 10, but everyone else. 2, 4, 5, 6, 7, 8, and 9. All those, some are positive, some are negative, but they're all absolute value-wise small, right? 2.5, 1.3, 2.5, 0.8, so forth. They're all relatively small. So delta G, whether it's positive or negative, as long as it's small, it indicates the reaction is reversible. If delta G is large, whether it's positive or negative, it's irreversible. And it'll only go in the direction in which it's large and negative. Right? That's why step 10 is only going to go in the forward direction because it's negative 16.7. But it's not going to go in the reverse direction. Whereas, let's say step 8, it's positive 0.8. So it can go either way rather easily. It doesn't really prefer one way or the other, not that much because it's a very small number. It doesn't matter if it's positive or negative. Right? Same thing with step 2. It actually prefers to go forward slightly, but it's virtually reversible. Whereas step 1 only prefers to go forward, just like 3 and 10. Fortunately, none of these have very large positive delta G's, which would mean it can't do that. It only goes the other way. Okay, so I don't want you to memorize this chart or anything like that. And if you tell yourself that little story again that we started with, you can look at the reaction type names there, and it reiterates that story. Putting a phosphate on, isomerizing it, putting another phosphate on, cleaving it into 2 molecules, and also all the way down. Tell yourself that little story. It'll remind you what's going on. Okay. So other than glucose, we eat other things besides glucose, right? With other sugars we talked about, such as galactose and lactose from milk, and fructose from fruits. So we have a lot of other sugars we can consume in our diet. So if you were to eat something other than glucose, like if you're eating something other than honey, right? So maybe you're drinking milk, right? You get lactose from milk. Milk lactose, remember it's a disaccharide. You can break it up into 2 sugars, right? They were galactose and glucose. You already know what to do with the glucose. What do we do with that galactose? Well, if you remember the structure of galactose, the only difference is the chirality difference between whether the OH on number 4 is on the left or the right. Remember, glucose was right, left, right, right. Well, if I switch number 4 around, instead of being on the right, it's on the left. We have right, left, left, right. That's galactose. Right? So the only difference here is I need an enzyme to convert galactose to glucose. Right? That'd be pretty easy to come up with. I need some kind of enzyme that would move that OH to the other side. Right? So it's an isomerase that'll do that. And then we can turn galactose into glucose. Pretty simple. And then you know what happens to it the rest of the way. The other sugar you can eat, a lot of it in fruits, right, is fructose. Well, all we have to do is phosphorylate the fructose. Right? We have an enzyme that can do that. In fact, it's the same enzyme. Right? Just put the phosphate directly on the fructose, and I have fructose 6-phosphate, and it's ready to be phosphorylated again. So rather easy to metabolize fructose. What about some other sugars like mannose? Right? Mannose is, again, it didn't make you memorize the structure of it, but it's another cousin of glucose where one of the OHs has been switched around. So I just need yet another isomerase. No problem. All right? What if you eat something like starch from potatoes? Right? So lots of sugars hooked together in a long polysaccharide. We can break those down into individual glucose molecules, or glucose with a phosphate on it, And we'll talk about that in a future lecture when we do glycogen metabolism. Right? So you can easily break those down as well. So lots of other places you can get sugars, but they all fork into the glycolysis pathway, one way or the other. Right. And lastly, normally I would have you, raise your hands in class. You can virtually raise your hands because I don't need to see the answers anyway. How many of you know that you're lactose intolerant? Right? If you have your hand up, you're a normal person, right? Those of us like me who are not lactose intolerant, we're mutants, right? Not the ones with the X-Men uniforms or anything, but we don't get any powers out of it. But we can drink lactose as adults, right? Or drink milk as adults. Those of you who are lactose intolerant, that's a normal operating protocol for mammals. You generally don't drink milk as adults, right? So being mammals, it's one of our three major features that makes us a mammal, is we produce milk for our young, right? That's where the name mammal comes from, mammary glands. So we make milk in the form of sugars like lactose in it, and you feed your newborns lactose. At some point, you're weaned off of the lactose, right? And the not newborn at this point, but the infant or toddler, or depending on the species, how old they are, they stop feeding off of the mother and start eating their own food. At some point, you got to make them do that, right? Because it's kind of a free meal up to that point. Now they have to forage for themselves, or kill something, or be fed by other individuals, or put their food in your mouth yourself, right? Whatever it takes, you got to get them off of those food and eat your own food. Well, a great way to do that is they can no longer digest the lactose. So the enzyme you use, called lactase, is made as an infant, and then as you start to get older, you start not producing it as much, right? The level of lactase production decreases, right? So way less than it was when you were born. So that last meal from mom didn't go so well, right? Because you couldn't digest it, and you spent your time in the bathroom or in the trees or whatever. I don't want to do that anymore. That wasn't very pleasant. So they get this feeling that maybe I should eat something different. That's how you wean them off of lactose, right? So for us, who are lactose tolerant, right, that happened in many places across the globe. That mutation developed where you still make lactase into adulthood. But one of the most common ancestral sources of it is in Northern Europe, right? So in Northern Europe, where the Vikings were, the precursors to the Vikings, in fact, so they had some harsh winters during the last ice age, and to survive the winter, you drank a lot of domesticated animals' milk, right? That's what they kept alive through the winter. If you couldn't do that, you didn't make it through the winter. So a lot of people who did not have the mutation didn't make it on to have children and grandchildren and so forth. So the popularity of the gene increased in the population. Those people are, descendants who became Vikings and Europeans and traveled most of the world now. That's why you see a lot of places don't have as high a lactose tolerance, or there's an increase in lactose intolerance, like in Asia and Southern Africa and so forth, in South America, some areas. But this is changing as we get, world travel and people settling and moving all over the place. Not so much right now, but that map is going to change where it's going to become uniform everywhere. That mutation developed in other places as well, but you can see why this has developed because we had an impetus to survive the winter. Most mammals don't have that, where they don't drink milk as adults. Now, some of you want to be physicians, and you move on to medical school, and you finish medical school, and you have some elderly patients, and the previous generations of doctors have been telling all those elderly patients their entire life to do what? What do they keep telling them their entire life to do? Right? And the goal of telling them this is to stave off osteoporosis. So lots of older physicians tell their patients to do this every day. And I've heard it from my grandmother many times.
Drink milk.
Drink your glass of milk every day, right? Have your glass of milk. Well, while it does stave off osteoporosis for a while, at some point, even those who are lactose tolerant, at some point, which this is not normal, right? Mammals aren't meant to live that old, right? Once you reproduce, this is a morbid thought, but once you reproduce biologically, you've done your job. So living into your 70s, 80s, 90s, even if you're lactose tolerant, at this point, the levels of lactase start to drop off dramatically. So you can no longer digest the milk, but they keep telling them to drink it to stave off that osteoporosis. So what happens is the galactose and the glucose aren't separated from each other, and there's a lot of lactose around. Well, there's some bacteria that eventually degrade the lactase or lactose for you, but galactose in the blood is not nearly as soluble as glucose, right? Just the way its OHs are conformation. Glucose is very soluble, galactose not so soluble in the blood. So when it's floating through the blood and goes through really fine capillaries, it tends to crystallize. So you've probably done this as a kid. You take some sugar solution, make it really rich in sugar, high concentration. You dip a string in it, you let the string dry out, and you get sugar crystals on the string, right? That's sucrose, but it's the same idea in the blood. Galactose is just not that soluble in the blood, so it tends to do this. So when it's going through really fine capillaries, right, the galactose tends to crystallize. And when it does it in the lens of the eye, right, or you can't see through it anymore, it's like looking through a stained glass window. And that's called cataracts. It's not the only source of cataracts, but it's one. It's galactosemia, too much galactose in the blood. Right? So elderly patients that continue to consume milk when they shouldn't, this is what happens. So today we have an easy fix to that. You can get milk without lactose, right? And you still have all the calcium you need to stave off the osteoporosis, but you don't need all the galactose that's going to cause cataracts. Right? That wasn't possible 200 years ago, but we can do it now. Right, so last thing I want to talk about is the regulation of glycolysis. So this should be common sense. You should easily pull this out from what we've said so far. If you want to regulate this pathway, you want to do it at steps that you have control over, that you can't allow to go backwards. Well, that's easy to do if you look for steps that are irreversible. Well, those are the three at the top, right? The hexokinase at number 1, PFK at number 3, and pyruvate kinase at number 10. Those three steps are irreversible. Look at those large negative delta Gs. So I want to control this pathway at those three steps. The first one, the hexokinase pathway, is pretty easy to regulate. When you make enough of its product, glucose 6-phosphate, it turns off the enzyme. This is simple negative feedback regulation. When the enzyme makes enough product, it turns off the enzyme so you don't make any more. Right? That's shown in the figure on the left. When you're at rest, you've got lots of energy, like you're listening to this lecture right now, sitting in your chair. You're not really doing a lot. You don't need to do glycolysis as much. So let's turn off the enzymes or turn them down a little bit. Okay? The other two enzymes, the phosphofructokinase and pyruvate kinase, who control steps 3 and 10, are more sensitive to the changes going on in the cell. Right? They're looking at, do we need to do glycolysis now? What would indicate, do I need to do this pathway or should I not do this pathway? What molecule in the cell do you think has bearing on that? So think about what does this pathway accomplish? What molecule in the cell might have an effect on whether we should do this or not?
Is it the mitochondria?
Well, so those are organelles. I'm looking for a molecule. In the cell.
Oxygen?
Not oxygen. What was the other thing you said?
ATP?
ATP, exactly. So one of the products of this pathway is ATP. And if I have an abundance of ATP, like when you're at rest and the cells aren't really doing much, then I shouldn't do this anymore. Let's not waste our glucose when we don't need it. So ATP in abundance will turn off enzymes numbers 3 and 10. So phosphofructokinase and pyruvate kinase both bind to ATP I know it's a normal part of their reaction is they bind ATP or ADP, but there's a second site where they bind another ATP and it turns it off. So if there's a lot of ATP around, it turns those two enzymes down or off. How would you detect if there isn't a lot of ATP? It's really hard to detect an absence of something. So how do you detect an absence of a molecule? In this case, we're trying to detect an absence of ATP. This is a little harder question. So it's really... Go ahead. It's really difficult to detect if something is not there. But it's easy to detect something that is there. So if there's not a lot of ATP around, there should be a lot of its alternate version, ADP or AMP, meaning the ATP without its phosphates, without all the phosphates. So what this enzyme does, PFK and pyruvate kinase, they both detect in the same spot AMP. If there's more AMP than ATP, we're clearly lacking in ATP. And that will turn this pathway on. Does that make sense? So if we have a lot of ATP, let's not make more. Let's turn this off. If we're lacking in ATP, there's going to be abundance of AMP, and that will turn this pathway on. Right, so that's all in this figure, and it should make logical sense why those things happen at those places. Okay? And I summed that up in the next slide as well here. Right, so I just wrote all those things out, I just said in words. So PFK is the most important control site. That's our step 3, the phosphofructokinase that turns the already phosphorylated fructose into the doubly phosphorylated fructose. ATP will inhibit it, and AMP will relieve the inhibition. Does that make sense? Hexokinase was at step 1. That one's really simple. It's inhibited by its own product. That's simple feedback inhibition. And we kind of let that one go to keep the glucose in the cell. And finally, the last one at step 10, very much like the phosphofructokinase, high ATP inhibits it, and a lack of ATP would not inhibit it. Right? So it's very easy to remember what does what here. So be able to explain what each of these does and why. All right, so what kind of questions do we have on this?